Thank you for joining today, everyone. We are excited to have our webinar today, Critical Considerations for Laboratory Sample Submissions. I'm excited to have Leo Schilling here with me today, hosting the webinar. I am Amy Knock, and I will be moderating today's session. Before we begin, I'll let you know more about how the webinar will run. The webinar is being recorded, and the slides and recording will be available for you within 3 business days. A short Q&A session will follow the presentation to answer any viewer-submitted questions that you may have. During the webinar, you can submit questions you have using the webinar sidebar menu. Select the Questions tab, type in your question, and then hit the Enter key on your keyboard. Remember, you can submit questions anytime. That concludes my brief introduction today. Leo, I will now pass it over to you to begin the webinar.
Thanks very much, Amy, and good afternoon, everyone. I'm Leo Schilling. I work for Eurofins SF Analytical, and I support the Animal Health Testing Laboratory. You could all join us today, and thanks for attending today's webinar. The discussions today are generally applicable to a wide variety of sample types, including food, feed ingredients, nutritional supplements, and botanicals. Developed for anybody interested in or involved in food or feed quality and safety. This presentation will review sample quality criteria and the parameters that should be considered in order to produce meaningful and defensible measurement data. A review of the purpose of standardized methods will also be presented, and considerations for when it's necessary to apply a modified procedure or an alternate method will also be discussed. So in today's presentation, I have it divided up into 5 sections. So on our agenda today, first I'm going to talk about the sample quality criteria. Next, I'll be reviewing some of the actions that take place in sample receiving. So that'll be rethinking sample receiving. So some thoughts for you guys to consider when you're sending samples. I'm going to briefly talk about rapid responses for field sampling and why that might be necessary. I'm also going to talk about selecting a correct analytical method for analysis of your sample, and then what to do when an applicable test method is not available. So, hi, I'm calling about your test method. That could either be you calling or me calling. No one likes a result that they're not expecting or a bad analytical result. So it's very important that we get a good sample for analysis, because we're making an inference. So an inference based on the analytical sample that we received is going to be the estimation of concentration or a characteristic of a larger amount of material from data derived from testing a smaller amount of material. So that's why it's critical that the sample that we receive is representative of the decision unit, and the decision unit in this case is going to be whatever the laboratory sample is representing, whether it's a tank of oil or a lot of soybeans or a truckload of grain. Inference to a decision unit is based on samples from the decision unit. So inference occurs at every mass production stage, from the analytical result to the decision unit in sampling to the testing pathway. So for example, inference from the analytical result to the test portion, or inference from the test portion to the analytical sample, or inference from the analytical sample to the primary sample, inference from the primary sample to decision unit, and so on. The number of inference steps is unique for each sampling project. So what's important to keep in mind here is that you're relying on the statistical inference when you submit a laboratory sample. So if the entire decision unit is not collected as the primary sample, and in most cases it is not, the portion selected from the decision unit needs to be representative to make inference back to the decision unit. These types of inferences are probabilistic inference and statistical inference. Consequently, the reliability of the test result is contingent on how well the laboratory sample represents the decision unit. So we're going to talk a little bit now about the sample quality criteria. So it's obviously very important that you establish good communication with all parties. So from the client side and from the laboratory side is critical. It is an advantage to clients, agencies, and industry to facilitate collection of representative samples, because unrepresentative samples may result in the wrong decision. So an example could be recall of a good lot of a product. So collaboration among all stakeholders is the best way to move forward to accomplish collection of a defensible sample. So if you ever have any questions about techniques that may be required for collecting a reliable and representative sample, always feel free to reach out and communicate with your laboratory about this. A long-term plan may take the form of periodic meetings between the client, compliance representative and laboratory personnel to agree on, the number of samples, identification of desired analytes, and logistics for submitting samples. Short-term planning, this could include situations like adverse events, where there's a violation investigation, maybe an unforeseen circumstance, or a foodborne pathogen outbreak. These should also occur at, to ensure efficient operations. This planning could involve discussion of sufficient personnel or surge capacity and expectations for turnaround time. And so, as you can see here on the slide, it's important that sample correctness is maintained, that, no biases are introduced, which means that, the same ratio of components in the laboratory sample, should be identical to as close as possible, identical as close as possible to the decision unit, and, reduce imprecision. So like I've mentioned before now, some important aspects of the decision unit is that, again, this is the bulk lot of the product. It needs to be accessible, so that when it's being sampled, a correct type of collection tools are used to take the samples are used, and also, to make sure, obviously, that the source is matched to the sample, so those aren't mismatched. So this is a super brief introduction to sampling theory. We could have a whole series on sampling theory, but just for the case of today, I'm just going to talk a little bit about two of the most important aspects. So minimum mass is the concept to control the error, essentially. It's important in the laboratory as it is in the collection of the primary sample. This is especially critical in the laboratory where very small test portion masses are taken for analysis, and the movement toward miniaturization, which is smaller test portions, essentially, smaller volumes of solvents and things like this, of analytical methods must consider minimum mass to control error. Although collecting random increments is generally considered the greatest challenge in the collection of the primary sample, insufficient mass is generally considered the greatest challenge in the laboratory. The consequences of sampling error are frequently ignored at many stages in the laboratory, because we assume that we have the best quality sample possible. So it's important to make sure that you're collecting a good quality sample so that the laboratory can produce a good quality result. Next, just going to talk a little bit about the number of increments. So the collection of multiple increments is critical in controlling distributional heterogeneity during the mass reduction step. One example of distributional heterogeneity is segregation of elements due to differing size, shape, and density. In the laboratory, collection of random increments is easy due to the small mass. It's accessible, and most laboratory samples are small, at least. The common practice of taking a single increment during mass reduction steps is not acceptable, and it's not defensible. So it's important to use the correct number of increments. There are very, very rare cases where a single increment is acceptable. So next, I'm just going to review some aspects of sample receiving that might not always be on the top of your mind when you're submitting a sample. So shipping conditions are very important concern. it's often an afterthought for a lot of companies, but if there are ever any questions about shipping conditions, you should certainly contact the right people to give you some advice on that. Storage conditions is related but not the same. Frequently after an analysis is completed, the laboratory will retain a portion of the sample potentially for retesting. So if the sample has specific storage requirements like low light levels or cold conditions, refrigerated or frozen, it's important to communicate that in sample documentation. It's also very important to keep in mind that if you're submitting a sample for multiple analyses, sample splitting might occur. So this image that I have up here is a rotary splitter, and it's designed to produce equal subsamples from the original sample, from the original laboratory sample. Also for most analyses, the samples are ground or comminuted, which is important to make sure that all of the analyte is accessible during the extraction process for the sample preparation. We often use specialized mixing equipment, and different types of samples require different types of mixing. It's also important to keep in mind that your sample is going to be distributed. So if there are qualities that your sample has that are vulnerable to transport segregation, it's important to keep in mind that you should make some notes about the properties of the sample or submit multiple samples to the laboratory. So I'm just going to talk a little bit more about each one of those items. So for shipping conditions, the ambient temperature during transport can vary, obviously, seasonally and regionally. So if normally sort of ambient conditions, shipping ambient conditions, but again, it's important to keep aware if it's going to a region where there's a lot of heat or cold. I mean, some products can't be frozen. There could be problems. Important to keep in mind that the conditions during shipping, have a sensitive sample might not be ideal. So you should consider using temperature-controlled packaging and insulated packaging. Storage conditions, as I mentioned before, are pretty critical because the laboratory without special instructions will store samples, usually at ambient conditions, and they may not be protected from light. So a lot of the analytes that are sent in for testing are sensitive to heat and light exposure. So if there are if there's some risk to losing the potential concentration, for something that has specific benefit or value in your sample, make sure to let us know in the documentation. And again, with subsampling, it's important to realize that there's an optimal amount of sample that the laboratory requires. There's also a minimal amount the laboratory requires. I would always default to the optimal amount. I wouldn't push the laboratory to use a minimum amount. I'll get into this a little bit later, but I mean, it really has to do with the distribution of the target analytes in the matrix. And so depending on the sample properties, a specific amount of sample has been validated for analysis. And if you contact your laboratory and ask them how much sample they need to send, they'll let you know. And so there's also kind of a question that you need to talk about with your laboratory if it's more appropriate to send replicate samples, duplicate samples, or if you would like the laboratory to split it. So comminution is necessary for most sample types, because it's important to make sure that the particle size is sufficiently small enough that the extraction solvents and other means that are used to expose the target analyte to the detector are available. So different methods require different sample preparation steps. So if you're sending in one sample for multiple analyses, you might consider sending in more than one sample because one method might require reduction to a specific particle size, where a different method might not require that level of particle size reduction and could actually also introduce some interferences that the test method wasn't developed to handle. So definitely important to make sure that if you're submitting samples for multiple analyses, to consider that the sample preparation might require multiple samples be submitted. Mixing is also a very important thing to keep in mind too, because there are different techniques for mixing, and some methods of mixing can actually promote segregation. So depending on the physical properties of the samples, some types of mixing could actually introduce a bias in results that could negatively impact the results. So if you have a sample matrix that is vulnerable to segregation, if there's a large variety of particle sizes in that, it would be beneficial for you and for the lab to communicate that with the laboratory when you're sending those samples. And finally, it's important to keep in mind too, when you do submit samples for multiple analyses, that the sample preparation involves maybe splitting down the sample into multiple samples, and it's going to be either transported to different areas of the laboratory or to even different sites. An example could be where a sample requires microbiological testing. And so in cases like this, the microbiological testing lab might not have the capability for running chemical analyses, and they might send a sub-portion or duplicate sample to another laboratory to complete the service. So, when you are dealing with multiple analyses, it's again important to understand which laboratory is doing what and what the best way to submit samples might be. And you can get advice from the lab for those topics. Again, taking another little deep dive into one of those aspects of sample receiving, and that's splitting. I just wanted to talk about some of the differences between liquid samples and solid samples, because even though this might be part of the activities that take place in the laboratory after a sample is received, this is also taking place when the client or the submitter is preparing the primary sample or the laboratory sample for submission. So the one exception that I mentioned earlier, where you could take a single increment, is with single phase liquids with no solids. In that case, it would be okay to pour or remove an increment with any kind of tool, because there's no separation. So it's uniform throughout. So that would be, I think, just about the only example that I can think of where it's okay to take one single increment. With multi-phasic liquids without solids and liquids with suspended solids, and slurries too, it's more than likely going to be best to sample those types of matrices with a cylindrical core. So some type of tube where you withdraw a sample to make sure that you're getting a nice even distribution that is identical to what would be in the sample. So again, you're just kind of like with the decision unit, the laboratory sample, you want to make sure that the subsamples are representative and have the same constituents and the same ratios as the sample that was taken from. So regarding solid samples, a stationary rifle type splitters consist of a hopper. I've got a picture of it up here, with a series of equal alternating inclined angle chutes or rifles, riffles, that empty into two receptacles. They can be gated or ungated. Stationary splitters are prone to operator error and misuse, and they can be used with care. But in rare in rare situations where distributional heterogeneity is low and the tolerable error is large, a splitter with fewer than 30 chutes may provide adequate split. But stationary splitters are inappropriate for most food applications. So this technique really only applies to flowable solids, like a grain or a feed. Rotary splitter is similar. I showed a picture of that, in a couple of slides back. So rotary splitter is a motorized mechanical device in which the sample is split into hundreds of increments. This device is very simple to operate. It's relatively inexpensive compared to other laboratory equipment. A rotary splitter technique is not as operator dependent as a stationary rifle splitting, but care still needs to be exercised. Rotary splitting is the most accurate because it selects more increments than any other technique. Finally, another technique is there's really three categories for, roughly speaking, for categories of splitting solid samples, and the final one is this fractional shoveling technique. And the size or mass of each increment is determined by the number of splits to be made. It also depends on the initial mass. The number of increments needed for each split it's really determined by, what your outcome is going to be. But when you're taking increments, you wouldn't just fill up the first split, from the primary sample and then go to the next. You would distribute each increment sequentially to each of the splits. The increment size must be reduced so that the finds are equally apportioned among all the splits. There are some advantages to fractional shoveling. It can be implemented in the laboratory or field. It doesn't require extra equipment. It has minimal cleanup and decontamination requirements, and any number of splits can be generated. But the important thing to remember, with the fractional shoveling technique is, that the entire sample should be increments should be taken from the entire sample. So if you have a very large area, it would be important to not concentrate the increments in one small part of that space, but to spread it out evenly over 2 dimensions and pull out increments from, in that way to create the split samples. So next, I'm just going to, kind of go through a couple of different sample type classifications. We get a huge variety of sample types. There's a huge variety of food products, everything in the grocery store you can think of. So they're designed to be shelf stable at ambient conditions, but they might require refrigerator or frozen conditions. So that's something important to talk to your laboratory about. We also receive beverages. So like I mentioned in that previous slide, the technique for sampling these could be different. If there's suspended solids in the beverages, it's important for the laboratory to use a different technique. So, if you have an unfiltered beverage, make sure that that information is shared with the laboratory, particularly if it's in packaging where you might not see the particles or where it's not apparent. Feed samples are commonly received. The important thing to consider about feed samples and feed ingredients is that they typically don't have preservatives, at least ones that aren't packaged for commercial use. If they are packaged for commercial use, it's likely they might have some type of preservative. But when shipping these, it's again important to consider appropriate conditions for both transport and storage after they're at the laboratory. Infant formulas, there are different forms of these, but most of them are designed to be shelf stable. But again, depending on the region and the time of year, it might be appropriate to send these with cold packs and insulated packaging. Same thing goes for supplements. A lot of the target analytes in supplement products are vulnerable to accelerated degradation. So it's important to make sure that you're preserving the sample integrity by making sure that no excessive heat is exposed to the samples. And finally, with dietary ingredients, I would say pretty much falls in the same category as in terms of care and handling as infant formula and supplements. It's possible that a lot of those materials might not have preservatives, and it's important to maintain the sample integrity, so it might be a good idea to ship those cold. So next, I'm just going to be talking a little bit about material properties here. So compositional heterogeneity is, the heterogeneity that is, arising from, just having different components. And, an example here is this bowl of nuts. So it's different particle sizes and different composition. Distributional heterogeneity is different. So you could get distributional heterogeneity from a sample matrix that you might consider uniform, but actually has different size particles and particles that have different densities. And this can be really tricky, particularly when when samples are sent to the laboratory, they're going through these transportation steps, and there's definitely vulnerability to transport segregation, where you'll have separation of fine particles and dense particles, and light particles and heavy particles. And this is very significant because this could require some special consideration on the part of the laboratory, both with comminution and with mixing. So very important to make notes like this to the laboratory that your product is not uniform, and so that we can take appropriate steps, because it could be possible that the analyte of interest is settling all the way to the bottom of the sample container, and if we are not aware of an appropriate way to or certain properties of that sample, it might not be mixed properly. So understanding the differences between compositional heterogeneity and distributional heterogeneity is very important. If you have any questions about that, always reach out to your laboratory or any one of the contacts that you think would have good information about this. And here is just a couple of really kind of take-home messages in kind of graphic format. In this first figure on the left side, you can see that the sampling tool is not appropriate for this sample matrix. The sample matrix could be a liquid or a solid in this case. But what's happening here is your sampling tool actually isn't large enough to capture some of the particles that are present in your sample. So you're absolutely going to get a biased result in this. And this could happen either with collecting that primary sample or laboratory sample, or it could also happen on after a sample is received by the laboratory. The figure here in the middle is kind of the same situation. You might have an appropriate tool for one type of matrix, but for another one, it is not. So this is a grain sampling probe. And what's happened with this sample is that it appears that there may have been some transport segregation where those either light and dense particles have separated, or the small and large particles have separated. But either way, it's important to see here that you're completely missing part of the sample formula or composition when you're taking the sample from the probe. So this would not be a representative sample. And finally, just kind of wanted to take a look at scoops. All scoops are not appropriate. Just because you can scoop up an amount of sample and you put it into a bag doesn't mean that it's being done properly. So you can see all of the scoops and shovels on the left side of these are curved, and the reason this is not good is because in cases where you have segregation of fine and large particle sizes, you're going to miss what's on the bottom. So the correct way of taking a sample in this case would be to use a flat scoop, and to make sure that you're taking this the entire column of a sample from the bottom. So you're capturing all distribution of particle sizes and densities. So how much sample is needed? It's a complicated question. So fundamental sampling error is imprecision error that's due to compositional heterogeneity, and that's kind of what we talked about before. You have different components. There's a reference that I'm going to give you at the end of this publication, this presentation, That's a really great reference for you guys if you want to take a deep dive into some of the theory and calculations that are involved with taking sample sizes. But really, the equation for variance for fundamental sampling error is to represent the analyte of interest, and it's shown here with the variables defined. Calculation of the FSC is useful when considering the appropriate amount of sample. So when the selected mass is less than 10% of the entire mass, the sample mass to control the variance of FSC can be calculated by using the relationship highlighted by the framed figure. So it's complicated. So really, the take-home message here is to ask your lab how much they need and make sure that you send the requested amount. To kind of wrap this up in a way that might be a little easier to understand, if you have I have actually a good example next. Let me just get to that. So this is like a case where we're going to the farthest end where you would need to submit the largest sample size, and so total mixed ration is a method of feeding dairy cattle. The purpose of feeding a TMR diet is that each cow can consume the required level of nutrients in each bite. So that's making the assumption that that it's well mixed. And a cow's ration should include good quality forages and a balance of grains and proteins and vitamins and minerals, just like a primary sample from a decision unit. So you can think of just each bite should be complete, just like a primary sample should be representative of the decision unit. TMR is a very challenging sample type for the analytical laboratory due to its high degree of compositional heterogeneity. You can think of it as feed composed of sticks, rocks, and leaves and twigs. Anything a cow would find out in the field when foraging for food. It also has high distributional heterogeneity, because there's probability for segregation of these components because they're different sizes and different densities. Particularly when a mineral premix or veterinary drug additives are part of the formula. Also, this material is very high in moisture, up to 60%, which requires cryo grinding and specialized milling equipment. So this is kind of the trifecta of difficult samples. So high distributional heterogeneity, high compositional heterogeneity, and high moisture. So to deal with this, we need to make sure that, since some of the components in this product might be heat sensitive, say in the case of, might have drug compounds in there, we would want to make sure that we're reducing friction. So because of the high moisture content in there, we would need to use cryo grinding. So cryo grinding just means that we're going to make the sample very, very cold, probably frozen. And that's going to build up excess accumulation of residues on the equipment, which is also going to reduce bias, because the residues that build up on the equipment could also contain critical components that are being analyzed in the analysis. So the solution here is to use a specialized piece of equipment. What I have pictured here is it's called a rotor beater mill. It has a 360-degree sieve. And there's a flow through attachment there. It's actually a vacuum attached to it that helps the sample flow out of the sieve after the particles have been reduced. So that helps to keep the temperature down and to eliminate buildup of the sample residue on the equipment. And then the result here is a nice powdery fine ground sample in the end versus a very clumpy, hard to mix, nearly impossible to mix sample if you used kind of a traditional mill or blender. So in summary, whether using online ordering or completing the sample analysis request form, it's critical that the laboratory receives all the information they need to properly handle a sample to produce an analytical result that's representative of the decision unit. So we have a double entendre here. This is a lab sample, but not a lab sample. So I took this photo from this reference here, this from AFCO Good Samples, and good test portions are two references that I'll encourage you to look up on your own after this presentation. So next, I just want to briefly touch on some situations that might require a rapid response for field sampling. So occasionally you might find yourself facing an adverse event that requires immediate response. It could be due to risk of health and safety for consumers. Reasons that a field sample might need to be collected could include suspected contamination with a foodborne pathogen or industrial contaminant, mycotoxin or heavy metal. Also, there could be a complaint, resulting, this suspected out of specification results, maybe an adulterated product or a counterfeit product, or disputes on nutrient claims. So the best way to do this, this is obviously, going to be something that needs to be handled very quickly. So the best way to deal with this is to make sure that you're deploying a regional qualified field sample technician. It's important that your technician is qualified because in a lot of these cases, particularly with a foodborne pathogen, or maybe the location where the product is being stored, if it's in a distribution warehouse or in a shipping container, or at a dock, it could be hazardous or dangerous. So making sure that your field sampling technician is qualified is critical. And here's just an example, just kind of a take-home message for you. There was a report of listeria contaminating a product that was made from soybeans. So, a regional field technician was deployed to collect samples, from remaining batches of the same lot of the product in the storage distribution center. And, what was discovered was that, that presence of listeria was confirmed. And so, what was important about this, we were able to prevent, continued contamination in other products with the same material. So this is one very good example of the importance of field sampling. Next, I'm going to be talking about selecting the correct analytical method of analysis. So we got once again a huge variety of samples. Food components can be one ingredient, or they can be a combination of many ingredients. It can be high moisture or low moisture. The same thing with beverages. It could be just one or two components or very complex. Feed and infant formulas are the same way. So what's important to keep in mind here is that there's a huge difference in between the methods of analysis that are used for high concentration products versus ones where you're looking for an analyte that is very low concentration. This is not by any means an all-inclusive list. You might also be dealing with environmental samples or drug products or medical devices. But the important thing to keep in mind here is that multiple methods are available for your analyte of interest. But selecting the applicable method depends on the physical properties of the sample and the need for compliance with regulatory agencies. So that might be another issue that you need to consider. So when particularly with regulated products or dealing with cross-border regulations or things like this, standard or compendial methods of analysis are preferred because these are methods that are standardized and performed identically in different laboratories. So the value of that is that a sample, conceivably a sample that's tested for one analyte in a laboratory could be directly comparable to a result that was generated by a different laboratory for the same or duplicate sample. So this is very critical, when you're using different suppliers for ingredients, or these areas of the world where there are maybe different growing conditions. So the concentration of the target ingredient might be different. So using standard methods is the preferred way to go, especially if the product is subject to regulatory requirements. In cases where you have a product that is new or a method is outdated, it might be important to have a internally developed or a special analysis performed on that sample. So a lot of the standard methods were designed for dietary ingredients or high concentration samples, and in those cases, you might not have a detector that is specific to that analyte. So if the formula changes, so if you're, let's say, manufacturer of essential oils, and you've been testing these, you've been having these essential oils tested by a laboratory, and your company decides to make a new product from those essential oils. It's important to communicate the difference in that formula and the presence of potential interferences with the laboratory, because it might be necessary to choose a different method of analysis because of those interferences that could be contributed by those different ingredients. So in those cases, we always recommend partnering with a laboratory that's specializes in the analysis of that category of products that you're using or having tested. And also there are a couple of different approaches for choosing a method. So I'm going to be talking about that now too. So occasionally there are times where your laboratory is going to tell you that the sample that you've just explained to them, you've given them all the details that I've just talked about for the past few minutes, and they tell you that they don't have a test method that's directly applicable. So what are your next steps? So for analytical method development, there's a couple of options. Option one, and this is the preferred approach, is to modify a standard or compendial method. And the idea here is you take a method that's mostly applicable, but just might need a small tweak to make it directly apply to your sample matrix. The advantages to this is that there's minimal modification, so there's going to be less costs, less work, and less time. Disadvantages are a lot of these methods are outdated. So we'll be restricted to using outdated technology a lot of times. So sometimes these methods of analysis are more costly because outdated equipment and supplies are difficult to maintain or obtain. So in cases like this, where you have the option to move to developing a new method, it might be preferred. So that would be the option two, developing an entirely new method. One of the biggest advantages of this is that it's going to be directly applicable to the sample matrix and the analyte, and it's going to utilize current technology. So you're going to end up with a highly specific method that's going to be developed to be as low cost as possible and efficient. There are some disadvantages. Obviously, it's going to take a longer time, take some time to research these things. Proof of concept experiments take time. So the process is very complex. Consequently, the cost can be very high. I just have one example here to kind of bring on the point, and this is a case where maybe an essential oil is manufactured from rosemary or thyme, and the high concentration method is suitable for the essential oil extract, but it's not suitable for the next stage product. So if you decide that you want to put this product in a fragrance or a soap or a feed, there are going to be interferences that weren't developed, that weren't considered when developing the original method. So in this case, we had to develop a new method. The analytes are volatile, so we use gas chromatography, and the original method used a flame ionization type detector. Unfortunately, it's not capable of resolving between co-eluding chromatographic peaks and other interferences. So we needed a method that was less vulnerable to to the new sample matrix. So the solution in that case was to develop an analytical procedure that minimally modified the original method. So it sounds like a huge change, but if we're maintaining the original extraction and just changing the detector, that actually is a significant amount of savings in terms of method development. So I still put this in the category of minimal modification. But the advantage here is that the new method is not vulnerable to interferences because it's detecting the analytes based on their molecular weight using mass spectrometry. Next, just want to talk about choosing the right partner. A lot of labs can commit to taking on a project, but making sure that you're sticking with a laboratory partner that specializes in the type of work that you need done is very important. Again, there are options for this. If your facility has a laboratory on site, you always have the option of internal method development. Some advantages to that obviously is that you're going to have lower cost. There's going to be less external communication. Trying to explain your products that your team might know inside and out, but sharing information about your product to an outside source might require a great deal of technical knowledge and experience. Communication could involve non-disclosure agreements and things like this. So there could be some reasons why you want to keep method development internal. Some disadvantages is it could extend a project timeline. You might have a heavy workload in your laboratory, and also a method development project takes a tremendous amount of project management. And another big risk is that internal methods are not always accepted by regulatory agencies, and this can be for a variety of reasons. Could be that method development plan wasn't robust. Maybe there wasn't enough testing of the procedure to make sure that it was robust. It could be in documentation, the way the report was collected, the way the data was managed. There are a lot of considerations. It could even be the lack of facility records on calibrations for equipment and things of that nature. So option two is choosing a CRO or a specialty research laboratory. And the big advantage to this is you have a team that's experienced in method development activities. So consequently, there's going to be less time and troubleshooting with research and experimental design. The trade-off here is that it's going to be higher cost. Other people that are very experienced are performing the work, and so this is costly. It also requires very good communication. So just because you're outsourcing this to a CRO or another research laboratory doesn't mean you're off the hook. You're still going to have somebody very technical in your organization to communicate and stay alongside of your laboratory partner through that method development process to make sure that it's meeting all the requirements for your purposes. And just kind of to bring home the point here, I have another case study example. Probably a lot of people are still familiar with the 2008 catastrophe of melamine. It's widely used industrial chemical. It's not considered acutely toxic with a high lethal dose in animals. And there was the 2008 outbreak in infants that showed that melamine ingested in large doses can cause kidney stones and illness without significant ingestion of cyanuric acid and other melamine-related compounds. So how did this happen? So in crude protein analysis, nitrogen is used to quantify protein levels, and melamine is a nitrogen-rich compound. It can't be differentiated from protein by some of the typical methods that are used for protein analysis. So, the laboratory doesn't see what it's not looking for. So in this case, melamine is not a typical component for any food products. So it wasn't being looked for. So the response to this was, to develop an analytical method that's specific to the detection of melamine and related compounds. So in this case, a method was developed by the FDA in partnership with some other teams to produce an LC-MS method that was adapted for the residues that was used for a feed method, actually. So again, this is another important example where specificity is important, where when your formula changes, it's important that the laboratory knows about it. So we're getting close to the end here, and one more thing that I wanted to talk about today with you folks who might be interested is, when you have a need for a new analytical method, who can you trust? And so that's going to be the topic of next webinar that I'm going to be co-presenting with Josh Ryan, who's the business unit manager for Eurofins SF Analytical in New Berlin, Wisconsin. He's got a lot of technical expertise, so I'm looking forward to that. The date is still to be determined, so if you need some information about that, you can contact us at info@eurofins.com. Also, some acknowledgements here. Chuck Ramsey, subject matter expert, sampling guru, taught me a lot of the things that I know about sampling theory, along with Nancy Tix, who is the workgroup chair for these publications that are free and available for download, and I encourage you to take a look at these. Again, there were many contributors that assisted Nancy and Chuck with, and these publications are developed by a working group that were led by Nancy, and they were established under a cooperative group between members of the Association of Public Health Laboratories, Association of Food and Drug Officials, and the Association of American Feed Control Officials and industry partners. So this is these two publications are extremely valuable, put together by a lot of smart people. So again, I encourage you to take a deep dive if you have interest to learn more. And at this time, I could take some questions if any come up.
Wonderful. Thanks so much for the presentation, Leo. I am just going to go ahead and launch the poll here. We always like to ask our participants if they would like someone to follow up with us after the presentation. So go ahead and submit your response here. And then we just had a couple of questions come into the chat. So the first question here is it better to send 2 samples for 2 different analyses or let the laboratory split it?
That's a great question. I briefly mentioned that earlier. The first example that comes to mind is microbiological analysis. So if there's a sample that needs micro testing and also needs chemistry analysis, that could be nutritional or a specialty analysis. If sufficient samples available and you have a high degree of confidence in collecting duplicate samples, again, kind of going back to some things that I talked about in the presentation, where splitting and subsampling requires that identical subsamples are produced. If you have a good degree of certainty that you're able to do that, I would recommend sending 2 samples, because it's possible that each laboratory could have an impact on the sample handling that could affect the result of the analysis at the end. So if it's possible to send 2 samples, I would do that. But I would definitely take the advice from the laboratory. So make sure that you really ask them their opinion on that.
Okay, great, thank you. And the other question that we had come in here, will the lab make recommendations for shipping conditions?
Certainly. Particularly for samples, that are vulnerable to heat and light and cold. Like I mentioned, there is potential for samples to freeze. So if there's a sample that could be damaged from a freeze-thaw cycle, insulated packaging might be appropriate for that. If it's if you're shipping out and it's -5 degrees and it's going to sit on the loading dock for a while. Similarly, if a sample is heat sensitive and you're sending that out in an uninsulated package without cold packs, if that's sitting in the sun and it's 100 degrees outside, it's going to be 150 degrees, inside of that package no time. So there's certainly some potential for analyte degradation there, and the issue with that is you no longer have a representative sample. So that laboratory sample is no longer representative of your decision unit. And so that's a pretty big risk. So having appropriate packaging is very critical.
Great, thank you. And it looks like we're all caught up with the questions in the chat. So again, I just want to remind everyone that a recording of the webinar along with a copy of the slides will be available within 3 business days. And thank you again, Leo, for your expertise and presentation today. And thanks everyone for attending.
Certainly. Thank you everyone for attending. Bye-bye.
Thank you. Bye.